Welcome to another episode of I Am Nana.
Using the I, and I am Nana. I'm your host, Irfani.
And I am your other host, Monica.
In today's live update, we will be talking about hemostats.
Oh, hemostats. Can you explain what hemostats are?
Yeah, so hemostats are used to control bleeding when someone is injured or undergoing surgery. They're really important to prevent excessive bleeding.
Oh, yes, that's a good thing to prevent for sure. And since these are in contact with the actual living tissue, they must be biocompatible, right?
Yeah, of course. And guess what? One of the most commonly used material for developing these hemostats can actually be derived naturally from chitin, which is a biopolymer, more specifically polysaccharides or sugars that are found in crab and shrimp shells as well as fungi cell walls.
Right. And that's why these compounds are biocompatible since they're derived from these biological resources. The chitin must be deacetylated, meaning that the acetyl groups are removed from the polymer itself. Isn't it cool that we can use these shells that we usually throw away after eating and they can be used for such an awesome reuseful purpose?
Yeah, well, we can't really reuse the ones that have been cooked because it's probably contaminated with all the spices. So yeah,
yeah, that's true.
Yeah. Anyway, so chitosan have many applications such as making permeable membranes and now hemostats. Now, the work today was conducted by scientists from Nankai University, and this work was published in Nature Communications. And they utilized 3D printing to make a hemostat that can rapidly stop blood and promote tissue healing.
And so we've previously actually talked about 3D printing meat using hydrogels, right? And in here, it is a similar technique, and it's used to make a 3D template of the sacrificial microchannels made of polylactic acid. These microchannels are sacrificial because they are removable, and their structure will become the empty spaces in the 3D structured material that we're printing.
Yes, so after they make the sacrificial microchannels, they poured the chitosan solutions and treat them under a process called lyophilization or freeze drying. And this happens when the water in solution freezes and then the solid ice crystals is converted to gas without it being converted back to water. And so when chitosan undergoes this process, the structure becomes very porous, like a sponge.
Right. Imagine the final product to be a spongy structure with microchannels. Now, the goal is to make a hemostat that can stop bleeding, right?
Mm-hmm.
That means the blood can enter this spongy structure, build up, and the coagulation factors in the blood will actually promote blood clotting. And to better promote the coagulation properties, alkyl groups, which are hydrophobic CH3 or methyl groups, are added to further promote coagulation and prevent infection.
Mm-hmm. Okay, so let's start with how the fabrication process influences the mechanical structure of the sponge. There are two main factors that must be considered, the chitosan concentration and the PLA microchannel filling and diameter. So they found that 4% chitosan was sufficient for the structure to maintain its overall structure, which is important to prevent deformation when it's placed in the tissue and the external stress that's exerted onto the chitosan. Then they also found that the microchannel PLA filling ratio increases. So that means how much you're actually adding PLA into the structure. The porosity increases, which is as expected. Now, for the microchannel diameter, they found that the compressive stress, which is a measure of how much compression a material can handle before it deforms, actually decreases as the diameter increases. But when it's soaked with blood, these blood clots within these microchannels that was formed from the PLA, and it can actually help to prevent the deformation. They finally found that the best chitosan hemostat is the one that has a high porosity and large surface area. And the alkyl groups that was mentioned earlier also improves blood clotting compared to the ones that didn't have it.
Right. So the hemostat's mechanical integrity is highly dependent on its ability to essentially allow blood to build up and clot. Now, we need the hemostat to essentially mold itself within the injury site, right? That
Mhm.
means that it has to expand when it absorbs blood. I mean, it's very rare for an injury to be a perfect square or a
Yeah,
circle.
that never happens.
They come in all different shapes and sizes. So, since the hemostat has the microchannels and is highly porous, water and blood can quickly enter and expand to stop bleeding in the injury site. Now, the microchannels are really important since the ones without them, the hemostats without these microchannels, do not absorb blood as fast.
So the microchannels are acting like a highway for blood flow.
Yeah,
Yeah.
exactly. And when you squeeze out the water or blood from them, they are actually able to recover to their original shape. That's amazing.
Wow. Shape-shifting,
Yeah.
shape-shifting material. So after they characterize all of the properties of this hemostat, they confirm that the hemostat have better procoagulation performance compared to some of the commercial gauzes. And when they tested the hemostat on mice and pigs to mimic the hemorrhaging during surgery and by measuring how much bleeding was actually observed in the tissue after the hemostat was placed in the wounds, they found that the hemostats prevented blood loss more than the commercial hemostat often used in hospitals. And they also have a fast response time of 2 minutes compared to the commercial one at around 9 minutes.
That's amazing. That is really great. Very impressive, especially when there is excessive bleeding. Now, bacterial studies were also conducted, and in these studies, it was found that the hemostat did not show significant bacterial growth compared to commercial products and gauzes. The researchers also observed that the hemostat is compatible to the tissue and the blood cells. This is great because once it's in there, you have to make sure that it won't cause any infection. And what is most interesting is that these hemostats can be left in the injury site and the surrounding tissue can enter to start regenerating the tissue that was injured and
Yeah.
destroyed over the process. That's amazing.
Yeah, for sure. Yeah, I mean, again, it will prevent unnecessary bleeding and the fact that it can,
Mhm.
the tissue can start regenerating within the chitosan is amazing.
Mhm.
And just think how life-saving this will be.
Yeah. Yeah.
First responders should really have these on hand.
Oh, agreed, 100% for sure. But also, I'll add that the challenge here is the 3D printing process itself. How can it be scalable for large production? That needs to be addressed, and it takes quite a bit of time to 3D print something. It's always not so easy as they show in these YouTube videos. Like it takes a while, and maybe we could have a bigger 3D printer, like the ones that they use to make houses. Maybe that could work.
Yeah. Well, maybe that one's a bit too big for somewhere. That one,
Yeah.
the ones in the lab can definitely make it work. All right, that is all the Nano Tips today. Take care
And stay curious.